All right. Good morning, you guys. This is Sunday, the 23rd. So this is the day before our class actually starts that I'm recording this lecture. I'm looking forward to starting the semester tomorrow. Obviously, you guys aren't going to see this video for a couple weeks. This is one of my favorite lectures, simply because you're going to get this information again if you ever take a nutrition class. So this is essentially like a very high level nutrition class. We're going to talk about all of the molecules, which if you remember from our previous chemistry lecture, a molecule is simply just an arrangement of atoms that are covalently bonded to each other. And so there's certain arrangements of covalent bonds of certain elements, especially the ones that include carbon, nitrogen, oxygen, and hydrogen, that all come together to form these four categories of macromolecules, is what I call them in the note sheet. But they are biological molecules, and so they are the building blocks of the bigger biological molecules. And so I'm excited to share this with you. So let's jump into Google Chrome here. Sharing my screen. Just got to move my tab over. Nope. There we go. All right, so here we are in Canvas. So I'm just going to slide down. And this is a lecture that I've intended to be watched week 3. So you got a couple of weeks before you're going to see this. I'm going to click on the slideshow. And then I'm going to go to the actual slide page. And again, feel free. So it says open with. I'm just going to tell it's open with Google Slides. And so feel free to make copies of these slideshows, include them in your own Google Drive if you'd like. Feel free to write notes into the margins. Yeah, but let's get started. So there are 4 categories of biomolecules. There are fats, carbohydrates, proteins, and nucleic acids. You might say, like, wait a second, the slide says lipids. A lipid is a fat. They are exactly the same thing. And so when we talk about lipids, which is the first thing we start off with, lipids are fats, oils, esters. And then if you look down here, this molecule is actually cholesterol. And so esters would be things like waxes. So like honeycomb, beeswax, candles, that sort of stuff is an ester, and that's considered a fat or an oil. And then you have, if you notice here, they look at these molecules with all of their carbons and hydrogens attached. And so we're going to talk about fats next. And what we do is we step into this conversation and we start off with looking at what is like the monomer unit, the monomer subunit of a fat, and then how are those arranged together to make a polymer. And let me grab my pointer. And this molecule right here is a triglyceride. So if you've ever gone to the doctor and had blood work done and they do triglyceride counts, they're testing the presence of these molecules here. And you might look at this thing and be like, holy cow, that is a lot of carbons and hydrogens attached. Well, this unit right here, this carbon double bonded to the oxygen, single bonded to that oxygen, this molecule right here is the monomer subunit of a fat, of a fat polymer. And so we're going to go into detail on what that looks like. So let's just start off with the basics. The monomer of a lipid is called a fatty acid. Now there are 2 types of fatty acids. Okay. One is a saturated fat, and that's one where all of the bonds of carbon are taken up individually by hydrogens. And then there are unsaturated fats, for which there are many different types. And you'll see why there are so many different types of unsaturated fats. So let's take a moment here and just look at the structure of a fatty acid. And so we have a carboxylic acid, right? That's why it's called a fatty acid. Remember this functional group from our previous lecture? I told you, I try not to tell you too much information that does not get repeated. So most of the information we see or that I present is going to be repeated at a later time. So this is an example of that. This is a carboxylic acid. Remember I said the hydrogen can pop off and leave its electron behind, which makes this an acid. The rest of this, so let me see. This thing keeps popping up. I don't know if you can see it on your screen. There we go. Okay. Let's talk about its structure. So it's got the carboxylic acid head, and then it's got what we call a hydrocarbon tail. And so a hydrocarbon tail is simply a bunch of carbons single bonded to each other in a chain, and then the rest of their bonds are filled out by hydrogens. And so this particular fatty acid is what we call a saturated fatty acid. Now, there are different types, like I said, of fatty acids. There's saturated and unsaturated. And so this is a little bit of a misnomer. Hold on a second. I'm going to change something there. I saw something on the slide I did not like, and so I had to change it. So I used to say all 4 bonds of each carbon are saturated with hydrogen, but that doesn't make sense because if we go back here, right, not all 4 bonds of this carbon are saturated with hydrogens, but all the remaining bonds are. So you have carbon single bonded to carbon single bonded to carbon all the way down. It's the remaining bonds that are all saturated with hydrogens. So I misspoke there where in the slide where it said 4 bonds of carbon are saturated. Okay. And this is an example of a saturated fat right here. It's got, I want to show you something with the structure of these things. It looks complicated. It looks complex when you look at this structure here. You get the zigzags, and it makes people's eyes kind of gloss over because they're like, oh, that's like advanced chemistry stuff. But honestly, like, it's shorthand and it's very simple. Anywhere you see an intersection of lines, like these 2 lines coming down to this triangle, like there's essentially a triangle happening here. You've got a line coming from oxygen, 2 coming from the other oxygen, and then another line coming from somewhere else. Anytime you see an intersection of lines, that represents 1 carbon atom. Anytime you see a kink, or kink is not the right word. Anytime you see a bend or a pivot along the chain, at that point is another carbon. So each one of these points is a carbon. And what I did was I actually made this molecule here into the shorthand here. And I did that on purpose so we can count the carbons and you can see how these 2 things are related. This image here is a Lewis diagram of a fatty acid. And what we'll do is we'll start from the tail and count towards the head, the carboxylic head. So we've got 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. We've got 10 carbons in this fatty acid. Now, if we start at the tail here, you'll notice that it's a mirror image, right? This molecule here has just been flipped 180 degrees, but it's still the same molecule. And what we can do is we can count here. So there's a point here, and that's got a carbon. So that's 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. So this shorthand molecule is the same molecule as this one down here. And so you'll see them drawn 2 different ways. Notice this one has like an H drawn onto that O, whereas this one has a minus sign. All that means is that this H is popped off on this molecule, and it's now that hydrogen proton is now floating around as an acid. Okay. So like I said, that's what saturated fats are. Now, in unsaturated fats, of which there are many types, unsaturated fats, some of the bonds between the carbon atoms themselves are double bonds. So it's like the 2 carbons next to each other are not only sharing 1 bond with each other, they're sharing an entire second bond with each other. And what that does is it changes the angle of the molecule. It puts a kink in the tail. So you'll see in this one, if you count 1, 2, 3 back from the end, you've got this double bond here between the 3rd and 4th carbon. And what that does is it changes the molecular structure. It just changes the molecular geometry of it. It adds a little bit of an angle to the hydrocarbon tail. Whereas if you look at this saturated one up here, it's a nice zigzag straight line like a Charlie Brown t-shirt. Whereas this one is not going to have that nice straight line. It's going to have kinks. And so let's talk a little bit about saturated fats, because these are the ones that are kind of fun. So these double bonds have kinks in them. And the more unsaturated they are, the more kinks they have, the more double bonds they have. And what you can imagine is that a saturated fat, because it has such a nice uniform geometry, if that molecule is placed next to another saturated fat, those kinks will all, or the bends will all line up, and those 2 molecules will be able to get closer together. Where now, if we go back to our van der Waal interaction from the chemistry lecture, remember there's instantaneous dipoles happening between the carbons and the hydrogens in this hydrocarbon tail. And those instantaneous dipoles of the electrons being passed back and forth cause those two molecules, the two saturated fats, to zip together, to be attracted to each other like two magnets. And when they are attracted to each other and they zip together and they hug each other like that, they become a solid. Like the state of the matter becomes a solid. So it goes from liquid to solid. And the only way to melt it is to add heat to it, to add vibration to it that breaks those magnets apart, and then they can separate. But if you have unsaturated fats and they've got these weird kinky tails and they can't get close enough to get together, to zip together like magnets, then it's going to require you to cool off the temperature so much more that they can finally slow down enough to find some attraction between each other and become a solid. And so what you end up having happen here is that saturated fats have much higher melting points. That's what the MP stands for. Much higher melting points, meaning that they'll be a solid at much higher temperatures. It requires you to add more heat energy to those molecules, to that bucket of molecules, to make them turn to a liquid, from a solid to a liquid. And you see this in, like, if you've ever bought coconut oil from the store during the summer, the grocery store is kept at like 72 degrees. And so the coconut oil sitting on the shelf is a nice rich solid. Coconut oil, as you know, is full of medium chain triglycerides, is full of saturated fats. And so it sits at room temperature in the grocery store as a solid. But when you bring it home and you bring it in your house, that, you know, maybe you're saving some AC money and your house is set at 82 degrees Fahrenheit and your coconut oil sitting on the top shelf in your pantry, guess what? That small addition of heat to that saturated fat in the coconut oil turns it from a solid to a liquid. And we see this, we can see this in sort of the opposite way with unsaturated fats. If you take something like olive oil, which has a lot of unsaturated fat, sits on the same shelf as the coconut oil and stays a liquid, and will not turn to a solid despite you cooling it off. And so if you take that coconut, or if you take that olive oil and put it in your fridge, it'll get cloudy, but it won't turn to a solid. It requires a much, much lower point to become a solid. And so I find that quite interesting. I'm also going to explain another part of this that I'm not testing you on, so it's not on the note sheet, but super fascinating about health in general. All right, and here's the fact. You've all heard of omega-3s, fatty acids like ALA, DHA, EPA, all that stuff, right? So unsaturated fats have got sort of this they've almost been, what's the word, oversimplified in their healthiness. And so you'll hear a lot like, oh, I can't eat saturated fat because it's bad for my body, but I can't eat unsaturated fat. And it's like, that's an oversimplification of the process. So one of the problems with unsaturated fats is that corn oil has a whole bunch of omega-6 fatty acids in it. And corn oil is used in so much stuff that we have an abundance or an overrepresentation in our diet of omega-6s. Now, omega-6s, if unchecked or imbalanced with omega-3, will cause inflammation of cardiac tissue, blood vessels, things like that. And you'll get, it's considered to be an inflammatory unsaturated fat. Now, you can balance it back out by consciously taking in more omega-3s to bring the ratio back. So what happened was, ancestrally, we had a 4-to-1 ratio of omega-6s to omega-3s in our diets. Now we have a 24-to-1 ratio of omega-6s to omega-3s. And that's why people are experiencing so much inflammatory problems in their diet. This is why you hear doctors say, oh, take omega-3 supplements. By taking omega-3 supplements, you're bringing that ratio back to where the golden ratio was in our ancestry, from omega-6 to omega-3. Now let me explain where we get those numbers from. So in omega-9, omega-6, omega-3, any of those fatty acids have to do with where the double bond is in the hydrocarbon tail. So let me show you an omega-9 fatty acid first. So remember, we always count the carbons from the very tip, from the end. So we've got 1, 2, 3, 4, 5, 6, 7, 8, 9. Wherever you get to a double bond, that first double bond, that's what it is. So that's an omega-9 fatty acid. Oleic acid probably comes from canola oil, right? Canoleic acid, canola oil. The second one is an omega-6,9 linoleic acid. 1, 2, 3, 4, 5, 6. So notice when I get to the 6th carbon, I've got a double bond. And so that's an omega-6. So if I keep counting 6, 7, 8, 9, okay, so it's an omega-6,9 fatty acid. And then finally, your alpha linoleic or your ALA fatty acid, linolenic. It's hard to say that. Alpha linolenic. 1, 2, 3. I only move 3 from the end and I get a double bond. 4, 5, 6. Oh, look, I got another double bond. 7, 8, 9. There's my third double bond. So that's my omega-3, 6, 9. And so I thought that was really interesting. It's not something I quiz you guys on or test you guys on, but it's something that's interesting because you hear a lot about it in mainstream media. You probably come into contact or have been exposed to the idea that omega-3 fatty acids are good for you. And that's true. Like you should, if you eat fried food, you should definitely take omega-3 fatty acids. That's going to help you out, help reduce inflammation. All right, let's move on and talk about, we already talked about what's causing the different melting points. So I'm going to skip these slides. And let's just get into to make polymers of a lipid. So what we have here is it's another sort of diagram of a fatty acid. Notice we have our carboxylic acid head here, and then we have our hydrocarbon tail. Now they've drawn the diagram slightly different than the Lewis model, which is shown here, where everything's kind of at square edges to each other, and they've done bends, if you will. Now the one thing to consider and remember is that all of these molecules have been smashed onto a screen that is two-dimensional, but what they really are three-dimensional objects. Okay, so in order to make a fat molecule, meaning like a fat polymer, what we need to do is we need to have a glycerol backbone. Now, glycerol is actually a carbohydrate. So for every carbon, there's going to be a water molecule present. So glycerol is an actual type of carbohydrate. Now, it's used as sort of a, it's sort of used as a spine, if you will. And the fatty acid is like a limb sticking off the spine. And so in order to attach the limb to the spine and make a polymer, we have to do a dehydration reaction. So notice the H and the OH are coming off of the two molecules respectively to make H2O. And so that's a dehydration reaction to go from a monomer to a polymer. And this is going to happen 3 times in the making of a triglyceride. So notice I've attached one fatty acid here in this image, and the next one I've already attached the other 2 through the dehydration reaction. So in that process, I've lost 3 water molecules. And because I'm going from monomer to polymer, I'm building, so it's an anabolic reaction, and it requires the input of energy, and so it's a endergonic reaction. Okay, moving on. Next we have the phospholipids. So this molecule itself just a little bit complicated at a first glance, but what you can notice right away is you can notice that we have the glycerol backbone right here. That's the 3-carbon backbone. It's the same as this 1, 2, 3. 1, 2, 3. Even notice here, this carbon is attached to 2 hydrogens. This carbon is attached to 2 hydrogens. This carbon is attached to 1 hydrogen. This carbon is attached to 1. And then this carbon is attached to 2 hydrogens. And this carbon is attached to 2 hydrogens, right? We all see that. Okay. Now the difference here is that instead of having 3 fatty acid tails coming off of it, it only has 2 fatty acid tails. And one thing I want you to notice is tell me which one of them is unsaturated. Take a second. Which one of these two fatty acid tails is an unsaturated fat? If you answered this one right here with the double bond, you are correct with the kink in the tail. Now you can see how that molecular geometry is changed by having a double bond there. Now notice there's not a third fatty acid tail. Instead, this last carbon here is bound to a phosphate group. I told you this one would pop back in. A phospholipid. That's where the name comes from. It's a phosphate group and a lipid. And then the phosphate group is attached to this this molecule that's attached to the phosphate group can be one of many. It's what we consider to be like an antenna. It's like sticking out into the area outside of the cell or sticking out into the cell itself. So one thing to consider, right, if we're going back a little bit, is that nonpolar molecules are hydrophobic. Nonpolar molecules are hydrophobic, meaning they do not like water. So this is a problem. If our cell membranes, which exist in our bodies, which are like 70% water, how does that work? How do our cell membranes, how are they made up of molecules that are water-fearing or hydrophobic? Well, the ingenious part of the construction of physics and nature is that if you have two layers of these phospholipids, meaning one layer on top where the hydrophobic tails are pointed down, and one layer on the bottom where the hydrophobic tails are pointed up, remember, like dissolves like. And so the hydrophobic tails of the top group and the bottom group, they'll dissolve into each other. And this phosphate group is very polar. Those oxygens are extremely electronegative. They're dragging a bunch of electrons up there. And so those electrons are attracted to the positive dipole of the water molecule. And so it's able to dissolve just fine in a cytoplasm or just fine in the extracellular matrix outside the cell. And so you have almost a Jekyll and Hyde component to a phospholipid. You have the water-loving phosphate group that's sticking outside the cell or into the cell from the top or bottom layer. That little egg shape represents the phosphate group. So it's sticking out into the water. And then you've got was that Dr. Jekyll? So the Mr. Hyde, or is it Dr. Jekyll? Yeah, Mr. Hyde, you know, the water-fearing hydrophobic tails are in the middle. And so you have a two-part molecule, and then you have two layers of that molecule, and the two parts that don't like water dissolve into each other, and that makes up the cell membrane. And so I've already spoken about this, so we're going to skip that slide. You can see this was all the stuff I just explained. You have the hydrophilic phosphate heads, the hydrophobic or non-polar hydrocarbon tails that dissolve into each other, and you can even see the chemistry of it right here, where you have the two tails actually like each other, and then you have the water molecules. And notice the artist did a good job here showing that, see the two hydrogen heads are the two hydrogens here, which would represent the positive end of the dipole for a water molecule, are attracted to the negatively charged phosphate group. So the artists themselves oriented the water molecules to show and represent that there is a magnetic attraction between the phosphate group and the water. And so that's why those ends are able to dissolve into the cytoplasm and the extracellular matrix, respectively. Now, this is an oversimplification. This image is an oversimplification of the cell membrane itself. We're going to talk more in detail about cell membrane and cell membrane functions in our next PowerPoint, but this is a better artistic representation of what is taking place. And you'll see there's the inside of the cell has all these rope-like structures, which would be considered cytoskeletal, so cell skeleton. They would be made out of fibrous proteins, so there's structural proteins involved in holding the cell up. And then you've got these like weird embedded egg-shaped oblong units. Those are transmembrane, so they span the length of a membrane, proteins or enzyme complexes. And some of them have receptors that stick off into the liquid that's outside the cell, and they wait for hormone signals. Some of them actually send out signals. Some of them are attached to these extracellular matrix bundles, these fibers. The cell itself that we're looking at is probably a fibroblast, and a fibroblast is something that creates connective tissue, like in your joints, in your ligaments and stuff. And this hyaluronic acid, dude, I'm going so deep right here. I'm sorry if you're like glossing over, but you take glucosamine chondroitin for joint health, right? And that glucosamine provides the building blocks for this extracellular matrix of this cell that's creating your soft tissue around your joints and stuff. And so that's just, some fun facts. Now, there's another type of lipid I want to talk about, and that's cholesterol. So cholesterol is considered a lipid. Now, remember, anytime you see a point, right, or an intersection of these lines or a pivot in the line itself, that represents a carbon atom. So you've got a couple carbon atoms out here that they physically drew in. They didn't have to. They could have actually just taken those off and left the points out there. But they drew them in anyway. You've got a carbon atom here, one here, another carbon atom here, one in the middle here, a carbon atom here, carbon atom here. Now, here's the thing. In these ring structures where they form these 5 and 6 carbon rings, each point in the ring structure is a carbon. And so what you have these huge organic compounds or organic molecules, like cholesterol is one of them, that is just a whole series of carbon rings and carbons all bonded to hydrogens. Now you have this this OH group or this hydroxyl group here that is often modified to make things like hormones, but I'm getting ahead of myself. So there's several functions of cholesterol. Cholesterol is sort of like the precursor. Now we've all heard of that cholesterol is bad for us, or cholesterol, you should reduce cholesterol in your diet. Well, the truth of the matter is that your cells can produce their own cholesterol. They can make their own cholesterol. In fact, when we get to our cell structures, the smooth endoplasmic reticulum that surrounds the nucleus of the cell is responsible for cholesterol production in certain cells, like the kidneys, or in the liver and the gonads. But again, I digress. There's 2 major functions of cholesterol. And one is that the cholesterol itself is integrated into that phospholipid bilayer. So remember that the hydrocarbon tails are nonpolar that dissolve into each other. Well, within those, sometimes you'll find cholesterol molecules stuck into those animal membranes, but only of animals, to help with stability. So they stabilize the cell membranes in animals, but they also provide a level of flexibility. So it's like it can bend and then return to the same shape. And this is good if you're an organism like an animal that needs to move. Like if you have to bend and flex and move your cells and stuff like that, you're going to need to have some flexibility, but also stability so you don't fall apart or lose shape. And so they both stabilize and add flexibility to the membrane. All right, so the second function is that it's used as a precursor to steroid hormones, just steroid hormones. Now, the effects of these hormones are very pronounced, especially when you start talking about the major ones. So remember, they're nonpolar, so they can dissolve through the membrane, no problem, through cell membranes. The interesting thing about that is that there's no regulatory mechanism that the cell can employ to slow down the onslaught of steroid hormones. Whatever's in your bloodstream that's a steroid hormone is going to go through the cell membrane, dissolve right through it, like dissolves like. It's going to go through the nuclear membrane and right to the DNA where it targets it. So it can dissolve through all that and target specific genes on the DNA strands themselves and start transcription and translation, things we'll talk about in Unit 3. All right. So like I said, it can be a precursor to very powerful hormones such as testosterone, estrogen, progesterone. Aldosterone is one that regulates salt production in the body. Progesterone, it's one that regulates the menstrual cycle. Cortisol is a stress hormone. All of those are lipid-based. And so that's essentially wraps up the lipid portion of this discussion. But we are going to move on with carbohydrates. Okay, the monomer, well, let's just talk about carbohydrates real quick. The structure of a carbohydrate is simply this. There's monomer carbohydrates, and then there's polymer carbohydrates. Simple sugars, like the sugars found in milk or the honey, or in sweets, in iced teas, in fruit. All of these sugars, if you will, are simple, meaning that they exist in monomer form in these items. When we build complex carbohydrates, when we build complex polymers of these monomers that are found in these sugars, we get things that you're familiar with, like starches in potatoes or corn, or even in grains and pasta, some vegetables, that sort of stuff. And so we typically think about complex carbohydrates as being fiber and starch. But let's start with the monomers. A term you may hear for carbohydrates that's the same is saccharide. Now, the formula for a carbohydrate is characterized by the general formula. Not all carbohydrates specifically adhere to this formula, but it's a good start. Is for every carbon, there will be one water molecule present. So if I have one carbon, so N is 1, then that means I would have 2 hydrogens and 1 oxygen, which 2 hydrogens and 1 oxygen is 1 water molecule. If I have 2, that goes for my N, like an N equals 2, then I would have 2 carbons, I'd have 4 hydrogens and 2 oxygens, so I would have 2 water molecules for the 2 carbons I have. And so that's the general formula for a carbohydrate. Now, the general formula can be for a simple sugar, but it can also be for the polymer itself. Now, the monomer subunits of carbohydrates can be any of the following. And I'm going to show you a really scary slide, but I only need you to know 4 of them, and I'll tell you which ones those are. So a triose sugar, or triose monomer, would be any carbohydrate monomer that has 3 carbons in it. We will see dihydroxyacetone phosphate. We will see that again. We will see dihydroxyacetone. We see that when we do metabolism. We won't see glycerin aldehyde, and we probably won't see glycerin aldehyde phosphate. I don't think we will. Tetrose would be 4 sugars. Pentose, 5 sugars. Now, there's a couple of pentose molecules I do want you to know. The first pentose molecule I want you to know is deoxyribose. Deoxyribose, and I want to show you the difference here. If you look up here, this is an aldehyde up here at the top, and so you've got this carbon. So this is the 1 carbon, and this is the 2 carbon. This is the 1 carbon of ribose. This is the 2 carbon of ribose. And now what I want to show you is this. Deoxyribose is the sugar that is used in the monomer of a nucleotide of DNA. Deoxyribose. Deoxy. I'm skipping ahead of myself. The 2 carbons missing that oxygen. Do you see how that oxygen's missing? There's an H there and there's an OH there, so that oxygen is gone. So deoxyribose is one of them. That's a pentose sugar. It has 5 carbons. And then ribose is the next one. I want you guys to know that one too, because that's an RNA. You got DNA and RNA. DNA uses deoxyribose. RNA uses ribose. And then hexose would be any sugar that has 6 carbons, and there's a billion of them. In fact, I don't even have their names on this page, but I do have them on the next page. So one of the hexose sugars I would like you to know is glucose. Now, when I say I'd like you to know it, I just need you to know that it has 6 carbons, and it forms a hexagonal ring. So it forms a 6-sided ring. 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, and the 6 carbon sticks up away from the ring. And then the oxygen completes the 6-sided ring. The other hexose sugar I want you to know is fructose. And I want you to notice that fructose also has 1 carbon, 2 carbon, 3 carbon, 4 carbon, 5 carbon, 6 carbon. Wait a second. So glucose and fructose have the same chemical formula. Interesting. We'll get back to that in a second. I have a secondary drawing of deoxyribose and ribose as they will appear in a nucleotide. And notice the deoxyribose is missing an OH group here, and the ribose actually has that O. So there's an oxygen missing from deoxyribose, which makes sense in the name. Okay, going back to glucose and fructose. If we were to count up all the carbons and count up all the oxygens and count up all the hydrogens, we would come to find out that they have the same chemical formula. Really? C6H12O6. Well, if they have the same chemical formula, why are they different? Well, notice, right, and this is something to think about, is that they have the same building blocks, the same atoms, but they're arranged in different structures. The fructose is arranged in a pentagonial structure, whereas the glucose is arranged in a hexagonial structure. And so we call them isomers of each other. Same chemical formula, different structure, different function. Okay. All right. So, quick question, review. I'm going to drill this one into your head. So I apologize for the repetitiveness of this. But if we're going from a monomer to a polymer, what type of reaction are we going to be doing? You can bet your butt that if I'm asking this question this many times, it's going to be on the test or quiz. A dehydration reaction. All right, so let me show you a dehydration reaction where we take two monomers and we make a polymer. Now, the polymer is just a disaccharide. It's just going to have two sugars in it. But guess what? Anything more than one, anything more than mono is actually technically a poly. And so we're making a polymer of two glucose molecules. Now, one glucose molecule is going to lose an OH group from its one carbon. The other glucose molecule is going to lose the H from its 1, 2, 3, 4 carbon. And so what ends up happening is you get a 1,4 glycosidic, so that has to do with sugar linkage. So you're removing a water molecule, you're dehydrating it. This probably requires the input of energy because it's anabolic and endergonic, it's building, and you remove the water molecule, and then boom, you've got this linkage of this oxygen over here to that carbon. Dink, dink. We can also show you the dehydration reaction. It happens at synthesis sucrose. Now, sucrose is table sugar. Table sugar is made up of glucose and fructose. And we can dehydrate it. We can take the OH away from the one carbon of glucose and take the H group away from the, what, 1, 2, 3, 4, 5. I believe it'd be the 5, but it's calling that. Oh, I see why. It's because this is flipped around. So this is actually the 1, and that's the 2. Okay, so this is the 1 carbon of fructose. This is the 2 carbon. So we're taking the H away from the 2 carbon, the OH away from the 1 carbon of glucose, and then zoop, we got water, and then the bonds are joined together through that oxygen, and they call this a 1,2 glycosidic linkage. Something to keep in mind, this linkage, this 1,2 glycosidic linkage, you have an enzyme in your body that breaks that down and separates these into the individual monomers that get absorbed in your bloodstream. When you eat high fructose corn syrup, what you are eating is you're eating pre-processed sucrose that has had this glycosidic linkage removed. It makes it more shelf stable. Bacteria have a hard time breaking this down if that glycosidic linkage is removed. And so that's why we do create high fructose corn syrup. It's to preserve the sugars longer. If you just had table sugar in there, it would be more susceptible to degradation and digestion by small microorganisms. And so by removing that linkage, we get the same components in the syrup. It's just more stable. And the whole crap about like high fructose corn syrup causes obesity is crap. It's not true. It's not. If you eat too much sugar, that can be a problem. Period. Whether it's high fructose or regular sugar, doesn't matter. But people have jumped on that bandwagon, just like people in the 90s jumped on the bandwagon of salt, or in the 80s of salt being related to hypertension or blood pressure. And it's not true. Like, salt is not correlated to high blood pressure, but yet you still get boomers that will refuse. They'll order their food at a restaurant and be like, I don't want salt on my stuff. You better not put any salt on that. Are you trying to kill me? And it's like, no, your other poor dietary choices have led to your high blood pressure, not your choice of using salt on your hamburger, your big juicy hamburger with extra cheese. Okay, I digress. Anyways, so when we're going from monomer to polymer, we can make a lot of polymer if we'd like. It's not just limited to 2 glucose molecules glued together, but it could be thousands of glucose molecules glued together. And that's really helpful if you're a cell and you need to store some extra sugar energy away, you can take and store it as glycogen. And so our muscle tissue, which requires a lot of ATP, a lot of energy to move our muscle tissues, when you use them a lot, whether you're sedentary or otherwise, you still use your muscles a lot to get up and go to the bathroom or reach for the remote. You need the glycogen. The glycogen is going to be a storage of glucose molecules in our cells. And so the glycogen that is found in liver cells and muscle tissues of animals is called glycogen. I just did some circular reasoning there. The carbohydrate polymer that is found in liver cells and muscle tissue of animals is called glycogen, and it's made from excess glucose in the bloodstream. So anytime that you eat a bunch of sugar and your pancreas detects it and it spits out some insulin, it opens up these portals in your cells that allows the glucose to be taken out of the bloodstream and put in the cell. Now, not all cells are going to need that much glucose, and so what they'll do is they'll store it as glycogen. And so glycogen can be used if you're doing like endurance sports and stuff too. You know, you've heard of like carb loading. Carb loading is a way of increasing your glycogen stores before a big performance, an endurance performance. All right, there's another carbohydrate polymer that's, let's talk about that for a second. There's another carbohydrate polymer that makes up the cell walls of plants. And this is indigestible, and this is where we get into fiber. So remember I said that there was the complex carbohydrates and fiber. So complex carbohydrates, starches and stuff can be broken down and used by our bodies, but fiber cannot. And so cellulose is indigestible fiber, plant fiber, that we can use to help push bolus through our intestines and, you know, help it for help move food along through digestion. So we need it. We need that fiber, but we cannot break it down and use its energy. The generic term for a carbohydrate polymer is starch. And often starches are referred to as complex carbohydrates interchangeably. And so you'll hear us a lot, you know, in biology, we'll switch them out like a magician, and it will sound like magic, linguistic magic, but it's really not. We're just substituting and using the one that sounds the best in the right context. Okay, so that's carbohydrates. So monomers are called monosaccharides. Polymers are called polysaccharides or starches. I've got some examples of monosaccharides, the ones I want you guys to know, those 4. Like, be familiar with them. If I say glucose, you're like, oh yeah, that's a 6-sided one. And then fructose, oh yeah, that's the 5-sided one. And ribose and deoxyribose, oh yeah, those are the ones used in DNA and RNA, and one of them is missing the oxygen group. Polysaccharides, I need you to know glycogen and cellulose. Amylose is another type of starch that's a complex carbohydrate that plants use. So amylose would be like when you eat whole wheat bread or something, or crackers or something like that. Amylose is the starch that's used to make like saltine crackers. And so what's really cool is you have an enzyme in your mouth called amylase, which will break down amylose into sweet sugars. And if you chew on something long enough, like chew on a cracker long enough in your mouth, it actually will turn sweet. Go ahead and try it if you have some saltines and some spare time on your hands. It's kind of an interesting experiment. Okay, let's carry on to proteins. So proteins, as you guys hear a lot, are like, oh, you gotta get your proteins in and blah, blah, blah. Partially true. Definitely proteins are an important staple of our diet, but you can also overindulge in proteins, and a high protein diet can often lead to colon cancer. No, I mean, not like long term, obviously, not like you go eat a big steak and you got butt cancer. That is not how it works. Okay. But if the bacteria that are in your gut, if they are exposed to an abundance of protein for too long, you can develop polyps and sometimes colon cancer and other problems. You can develop issues that are related to colon cancer too. They might not be as serious as colon cancer, but it could be from extra protein in your diet. And so really proteins are about getting the right balance of proteins. But let's talk about what proteins are broken down into. Okay, so the monomer of the protein is an amino acid. Now, typically we have about 20 different amino acids that are used interchangeably throughout biology. And so one thing, amino acids are interesting because they have like 3 functional groups attached to them. And then it's a little bit confusing because if you go back to my previous lecture about functional groups were always attached to the R. And in proteins, it's like flipped. It's like so confusing. I'm sorry about that, but that's just kind of like how they're drawn in. R if you really want like a full explanation, the R itself is really just represents to replace something. So R is like you can replace the R with a bigger molecule. And in the case of like functional groups that in chemistry where we're talking about like the amino group or the carboxyl group or the hydroxyl group, the OH or the methyl group, yeah, those are all attached to that Honda Civic, right? And in this case, we actually have 3 functional groups. One of them has an R. And the reason why the R functional group has an R here is because remember there's 20 different types of amino acids. And so instead of drawing 20 different molecules, we can draw one generic molecule that all of the amino acids share, which is what you see at the bottom of the page, and then substitute another molecule here in the R position that will give it that 1 through 20 characteristic. And I'm going to show you this on the next page. See this? This is all the amino acids. Crazy, right? Okay, let's back up and take a breath. Woo! All right. So the structure of each amino acid is this. You've got a central carbon. And remember, carbons like to form 4 bonds. So one of the bonds that carbons forms is to a carboxyl group or a COOH. And remember, the carboxyl group can often be like an acid. So this hydrogen can pop off, no problem. And then you have an amine group, an NH2. And so this nitrogen has a lone pair of electrons right here. And this proton can often pop off and come over here and behave as if it's attached to the nitrogen. It kind of like plays with its extra set of action figure electrons. Because remember, think about hydrogen, right? If hydrogen pops off and it's just a proton, it has no action figures. It is not happy. So it comes over here and nitrogen happens to have like a set of Batman and Robin action figures. And so this little proton pops off and comes over here and he's like, oh, sweet. He's like, can I play with these? And nitrogen, since nitrogen has like the Avengers action figures that it's playing with currently, it's like, yeah, go ahead. You can hang out with my Batman and Robin and play, have a little play circle of your own right here. And so we'll often see NH3. We'll see NH3 with a plus sign next to it. And that's because that proton is adding an extra positive charge over here. And then when it leaves behind its electron, you'll often see a negative charge over on this end. Okay. Let's not go too much deeper than that. Let's look at all of the different amino acids. Now, these amino acids, if you'll notice, have all been flipped. So the functional group is actually, this R group is actually pointed down. The R group is highlighted in the purple box. Is that right? No. The purple box, what am I talking about? Sorry, the basic structure is the purple box, right? So you got the carboxylic acid. See how there's the minus sign there? And then the plus sign over here with the NH3. And then the functional group's up here. So this structure is seen here. The R group is what's highlighted in the yellow box. And so what you'll see is like amino acid. So remember, there's 20 different amino acids that we're going to use. Amino acid 1 is really simple, and it's just got a hydrogen. Just an H is attached here where this R group is. The next one has a methyl group attached to it. So it's a CH3 right here on this group. The next one has a CH and then it's bonded to 2 other methyl groups. And so you can see these growing chains, these functional groups on these 1, 2, 3, 4, 5, 6, 7, 8, 9 amino acids. And you might say, okay, well, why did you put all of these amino acids into this box and why are they all shaded yellow? Well, they're all shaded yellow because they are non-polar functional groups. Well, that's really interesting. So you do have functional groups. Some are hydrophobic, which means they do not like water. So if you have a hydrophobic functional group, that means it's nonpolar. So these are all your nonpolar functional groups. And what does that mean? They don't like water. And so if they're all joined together in a chain, they're going to fold that chain, that protein chain, that polypeptide chain, they're going to fold it in such a way where they're all facing away from the cytoplasm of the cell. Now, some functional groups are actually hydrophilic, which means they do like water. They are polar. So polar and hydrophilic, water loving and polar. Remember, water is like a magnet. So this polar magnetic, these are all magnetic side chains. And remember, the side chain is one of these funny things that we do in biology. is that we call the functional group the side chain too. So side chain and functional groups are the same thing in the case of amino acids. So these ones, see how they have an OH group? Well, that OH group is going to be polar automatically because the oxygen is always going to hog the electron from hydrogen. Yeah, you'll see oxygen a lot in these polar ones. So look up here, you don't see any oxygens in the non-polar ones, but in the polar ones you see oxygen because oxygen is extremely electronegative and it takes in a lot of electrons from surrounding covalent bonds and makes a dipole. Okay, and then some amino acids are going to have functional groups that are acidic. So notice these ones have an H group that popped off. So that's a carboxylic acid group that's attached to this functional group, and the hydrogen has already popped off in this case, and it's left behind its electron. And so that proton's floating around, making the solution acidic. And then over here you have basic or positively charged, meaning that they can remove a hydrogen from solution and grab onto it. And the way they do that is, remember, nitrogen has that lone pair of electrons. Nitrogen has a lone pair of electrons, and so that lone pair will pull automatically a proton out of solution, a hydrogen proton out of solution, and reduce the acidity of that solution, thus making it more basic. Whew, I know that's a lot to do this lecture. Trust me, go back and listen to it like 5 times, and maybe it'll all stick. I know it's a lot. Okay. When we're going from monomer, these are all monomers of a protein. When we go from monomer to polymer, we need to do a dehydration reaction. Now, there's a directionality to how we build a polypeptide. There's a directionality to how we build a chain of amino acids together. And the way it works is that one end of the amino acid will bond to the opposite end of the next amino acid. And so we see that here we have 2 amino acids that are bonded together. You'll notice the carboxylic head of the one amino acid is bonded to the amino end of the next amino acid. And then what they're showing you here is that OH group from the carboxylic group on this amino acid is going to pop off along with one of the H's from the amino group on the next one and form water. And then the bond that forms between carbon and nitrogen, well, that becomes your peptide bond. So if you're ever looking at a complex polypeptide and they don't look this pretty, trust me, they do not look this pretty. Whenever you're looking at it, if you need to count the number of amino acids in that polypeptide, in that big structure, all you got to do is look for the carbon bonded to the nitrogen, double bonded to an oxygen. Now you'll notice this carbon is bonded to a nitrogen, right? But it's not double bonded to an oxygen. So anytime you see a carbon double bonded to an oxygen, single bonded to a nitrogen, that's a clue. That's the linkage between 2 individual monomers, and you should be able to count the number of amino acids using that strategy, that technique. And so when you dehydrate this reaction, it's endergonic or anabolic. It's building it, it's going to require some energy coming in from outside. The water leaves, and you have a new peptide bond forming. Okay, now we get a chance to talk about the bigger 3-dimensional structure of polypeptides. I cannot stress this enough, okay? This part right here is the most important part of all of biology. Because what you have is you have 20 amino acids, 20 unique individuals. How do I put this? Go back. 20 unique building blocks. So this is similar to saying like I have a Lego set with 20 different types of Lego blocks. And what you can build with those 20 different types of Lego blocks is infinite. It is. It's literally infinite. I mean, because just because you have 20 different types of Lego blocks, I could say like, well, you've got 1,000 of these 20 different types of Lego blocks. What are all the possible combinations? Well, that's literally the equation for that would be 20 to the 1,000th power. That's more than there are molecules in the universe. So the possibilities, the endless possibilities of orders and sequences of putting these things into a certain order on a polypeptide is what gives rise to this complexity of life that we see. I can't stress this enough. And so you'll notice this is what we call the primary structure of a polypeptide. Notice there's a directionality to it, is that there's an amino end. So going back to this slide, remember the amino end is the beginning of the polypeptide, and then we're adding on new monomers, or adding on new amino acids onto the carboxylic end. And so this long string, this long polypeptide or polymer of a protein is characterized by having an amino end and a carboxylic end. One end's gonna, it's like if you took cars on the freeway, bumper to bumper, right? And you had a stretch of cars that were like, that rear-ended each other. There would be the hood end of the collision, where the first car in the collision, right? The hood, and then all the other cars are smashed together. And then there would be the bumper end of the collision. And that's the idea of this strand, is that you have the hood end, and then as you add on all of these cars in this car crash, is that then you have the bumper end at the very end. And so there is a directionality to the polypeptide. Now, each one of these Lego blocks, remember, that we're building this thing out of, has a unique set of characteristics that gives it a sort of personality. Is it hydrophobic, or is that a hydrophilic one? Does it like water? Does it not like water? And those interactions are going to dictate how this long string goes from a, if you think about it, it's a two-dimensional object. Maybe you could even argue that it's a one-dimensional object, because it's linear, if you will. It's got to start in it's a one line, right? They have beads on a string, if you will. Is how do you go from a two-dimensional or a one-dimensional object into a three-dimensional object that's all folded up into this shape that has a unique function in a cell? And that's the crazy part. That's the part where, even if you know the order and sequence that amino acids happen in a polypeptide, there are things like temperature, pH, what else? Well, those are the only ones I can think of right off the top of my head. The temperature and pH that will affect how this bead of strings folds into itself. And so your body chemistry, like inside of your cells, is going to, if the pH is high, then it might change the way that structure folds. If your body temperature is low, that structure might change its configuration and shape and change its function. And so there's so many variables that can affect how this one-dimensional object turns into a three-dimensional object. And one of those fundamental principles is the order and sequence of hydrophobic and hydrophilic amino acids in this chain, along with acidic and basic ones. So you've got these building blocks with unique characteristics that will all interact together in a variety of ways to form a big three-dimensional object. Now, it does that in a series of steps. It doesn't do it just like, and it's done. It does it in a series of steps. So let's consider each of those steps. First step is that you have this order and sequence of amino acids. That is called the primary structure. Now, when the beads on the string interact with each other, they can form secondary structures like beta sheets and alpha helices. And so a beta sheet is just like, it's going from that string into a serpentine formation. So it's like all of the amino acids that run across the top are attracted to the amino acids that are running in serpentine back across. And then those are all attracted in serpentine to the ones just below it. And so it forms this like back and forth serpentine sheet. Or the amino acids start to wrap and rotate around each other, because every so often in a sort of radial pattern, you get amino acids that recycle themselves in terms of their position on the string, and then they will attract to each other and form this this rotating column that we call an alpha helice. And then, okay, those secondary structures like alpha helices and beta sheets, as well as the string, still the primary structure, which is the string. So you've got like, it'll look like this. It'll be like you've got a string and then an alpha helice, and then more string and then a beta sheet, and then more string and another alpha helice. And so the strings plus the alpha helices plus the beta sheets all interact together to form the tertiary structure, which is the overall three-dimensional model of that protein. Then tertiary structures can interact as subunits to a larger quaternary structure. This is like hemoglobin in your bloodstream. And those tertiary structures come together to form a larger supermolecule that has a quaternary structure that, that serves a particular function in the cell. And so all of these proteins, the one thing to walk away with this as is all of these proteins have a unique function in the cell. Some of these proteins, when we make them, they just turn into a cytoskeletal element. They hold the cell up. Some of these proteins, when we make them, they get packaged into little vessels that'll get released into the bloodstream that serve as hormone signals to other cells. Sometimes these proteins will turn into receptors in the membrane of the cell itself, like there'll be a signal molecule or an antenna dish, if you will. Some of these structures that are formed, these tertiary or quaternary structures, will actually digest certain bonds from the molecules that we ingest, that we eat. And so you have this symphonic harmony of all of these proteins working together to perform particular functions in the cell, that give the cell its overall life history, its life functions. And so it's kind of like mind-boggling to really wrap your mind around that, especially if you can connect this idea that the order and sequence of amino acids in this primary structure for a protein, the order and sequence of those amino acids is corollary to the order and sequence of nucleotides in DNA. That's so important to know. Okay, and that's where we were at. Nucleotides. All right, so the monomer of any nucleic acid is going to be called a nucleotide. So that could be a RNA nucleotide or a DNA nucleotide. All right, so an RNA nucleotide is going to have ribonucleic acid. And so it'll have a ribose sugar like this one that has an OH group on the 2' carbon. It will be connected to a nitrogenous base. It will be uracil and RNA. And if we're talking about DNA, it'll be thymine. Otherwise, everything else is the same. Everything else is identical. Okay. Ooh, there goes the dog. All right. DNA is typically double-stranded. It's usually, you got 2 strands and they zip together, and is only found in the nucleus of eukaryotic cells, but it's found in the cytoplasm of prokaryotic cells. Now, prokaryotic cells don't have a nucleus. They don't have a way of compartmentalizing their DNA like eukaryotic cells do. Animal, plants, fungi, multicellular, those are all eukaryotic cells, and they're able to compartmentalize their DNA, the repository of information on how to build a protein. And I just lost my train of thought. So the order and sequence, I spoke about this earlier, but I do want to reiterate it, is the order and sequence of nucleotides in the DNA. So as they appear, cytosine, thymine, adenine, uracil, or guanine, cytosine, thymine, adenine. So as this order and sequence of nucleotides appear, it is directly parallel to the order and sequence of the amino acids in the polypeptide that we spoke about, which is really interesting because now it's like you have a repository of information that's in a different language. It's in the language of nucleotides, but it can be directly translated into the language of amino acids, which actually form a functional structure, which is incredible. DNA is typically double-stranded. Okay, so RNA is typically single-stranded. So even though RNA is single-stranded and it has that OH group on its 2' carbon and it uses uracil instead of thymine, even though it's single-stranded, it serves more than one function. It can perform several tasks in the cell, whereas DNA is just the repository of information on how to build a protein. RNA can actually be a functional molecule itself. We'll talk more about that in module 3. One of the tasks of this RNA molecule is to carry the information that's housed in the DNA to a place where it can be translated into the information on how to make an amino acid or build a polypeptide. Interesting thing about RNAs is actually more transient because it has that OH group on its 2' carbon. Because this OH group is here, it actually makes it more susceptible to degradation than DNA. DNA, because it's missing the OH group on its 2' carbon, is way more stable than RNA. And so the two things I want you to walk away from this is DNA stores information on how to build a protein, or it also has the information on how to build an RNA molecule. The DNA is missing the OH group from the 2' carbon of the ribose, making it deoxyribose. It's more stable. It's found in the nucleus of eukaryotic cells and in the cytoplasm of prokaryotic cells. It's double-stranded. RNA is a single-stranded direct RNA copy of DNA. And so instead of thymine, it uses uracil. And instead of deoxyribose, it uses ribose sugar. But other than that, they're essentially identical. It can be functional in its structure itself, or it can be read and translated by a ribosome into a protein, into a polypeptide. And so that wraps up our conversation about macromolecules. So a little bit of a nutrition lesson, a little bit of some fun facts, a little bit of connecting it with things that you've already heard, possibly from other sources about nutrition, diet, and stuff like that. And hopefully some more clarity around those things. All right, we'll see you guys later.